Good morning to everyone. I hope you can hear me. It's a Thursday morning, 10 a.m. means it's Enjoy Science. And of course, I have to thanks to all of you who are here, because without you, the attendees, such webinars make no sense. Of course, I have to thanks to Giulio Draeta, Chief Scientific Officer, and also to Ignacio Vistuba, because this Enjoy Science now it's going through CSO Office and Translation and Molecular Pathology Department. To thanks to Emily Lang, because without her work, nothing can be done. And of course, mostly to thank to Dr. Marina Konopleva, because in spite of her very busy schedule, she found time, in fact, to enjoy science and tell us how she enjoys science. And in fact, it's a pleasure to introduce Marina, because I know her for quite a bit, and I can tell you that she's in fact a truly outstanding clinician scientist. She is trained in the beautiful city in St. Petersburg, and then basically she arrived in MD Anderson, and she went up all the levels till the level of professor. But what is the most important, she is a wonderful clinician, and she's also a wonderful scientist, because Marina is running a very heavy and successful research program and research laboratory. She is splitting her life between the life of patient and the life of scientist. And in fact, she published wonderful papers, and most important, she is leading very interesting and very successful clinical trials for patients. So if I think to Marina, I think to an extraordinarily successful clinician scientist who can translate the discoveries from the bench to the bedside of patients. And with this, I let Marina to take the show with something that is very close to my heart, because venetoclax, it's working anti-BCL2, because BCL2 is a target of 2 micrornates 15-16 that I was lucky to discover to be abnormal in cancer. And with this, Marina, please run the show, and thank you all to be here with us.
Thank you, George, for this very kind introduction. It's a pleasure to be here. I will take you through the journey that I started actually when I came to MD Anderson in 1996 to Michael Andreev's lab. So BCL2 was my first project, and we're still working on that. And we were very fortunate to take this forward into clinical trials, which turned out to be quite successful. So I'll show you briefly some clinical data, and but mainly focusing on the lab and what we're doing today. A lot of the data I'll show you today are still unpublished. So thank you for being here this morning. These are my disclosures. So why is it important to work on BCL2 and target BCL2? As you're probably very well aware, the resistance to cell death is a hallmark of cancer, which has been identified quite a long time ago, and is a primary reason for resistance to the chemotherapy. So the fate of the cells as far as survival is largely controlled by the BCL2 family of the proteins, which contain both anti-apoptotic and pro-apoptotic family members. So BCL2 is right here, is one of the central family members. And if you look at the structure, so there are these BH domains that you can see very prominently in both anti and pro-apoptotic family members. And there's this domain called BH3, which is shared amongst different family members. From the pro-apoptotic, there are two large families. One are BACs, and BACs are called executioners of cell death. And then there's a large family of BH3 only proteins, which largely functions through negating the role of anti-apoptotic family members. So how does it work? So there's a certain hierarchy in the binding of the anti and pro-apoptotic family member. For example, here BCL2, it binds not to all BH3 only proteins, but it binds to BAD, BIM, and also actually it binds to BACs, which we showed back in 1996 when we published, in 2006, I'm sorry, when we published the first paper on the ABT737, the first BH3 mimetic. There are several, 5 family members of the anti-apoptotic proteins, and the ones I will talk to today is BCL-XL, a little bit of MCL1. So BCL-XL binds to other family members, and MCL1 is quite different. It shares the pro-apoptotic binding with the NOXA-BIM with another family member called A1 and also BAC. So essentially what happens is that the binding is executed through this BH3 domain that I mentioned on my previous slide. And so here, let's say there is BCO2 bound to BIM, which is BH3 only protein, is not activator. But when you have so-called BH3 mimetic, which is a small molecule mimicking BH3 peptide that binds specifically to BCO2, what happens, it will release the pro-apoptotic family member BIM that then will go ahead and bind directly to the bags and result in actually bags dimerization, which will go ahead and make pores on the outer mitochondrial membrane, resulting in cytochrome C release, caspase activation, and cell death. So I'll talk to you about this concept of BH3 mimetic and the drugs that went, as I mentioned, all the way to clinical trials. So this is the main drug I'll be discussing today. Inhibitor of BCO2 is a selective BCO2 inhibitor. The original name is ABT199. It was designed by AbbVie. It was published in 2013. It has high selectivity for BCO2. I mentioned to you the prior inhibitor, which was a first tool inhibitor, ABT737, that we published as has high activity against AML cells. So that was 10 years prior to the ABT199 discovery. So why that compound never really made it to clinic? Because BCL-XL is very important for platelet production. And therefore, the BCO2 BCL-XL inhibitors, while quite effective, they cause thrombocytopenia, on-target thrombocytopenia, which of course prohibited its development in acute myeloid leukemia setting. But I will get back to you on BCL-XL and show that it is still a very important target, and we have new tools to address that. So this is venetoclax, and again, it binds specifically in this pocket, which is BH3 pocket. It's actually composed of 3 different domains. And this is a structure, is a large molecule. So when the company came up with this inhibitor, essentially my lab, Dr. Jeff's lab, and Tony Lutai's lab has joined forces, and we decided to work on this together in acute myeloid leukemia because we knew from the prior work that BCO2 is highly important in AML, although perhaps not through microRNA, but through other mechanisms. So now I'll move on into preclinical studies. So this will be outline of my talk. I will discuss with you some preclinical data, but very briefly because it's published. And then I will move into new areas, how we address mechanisms of resistance and sensitivity, how we combine BCO2 inhibition and targeting immune system. And finally, what is the role of BCL-XL and how we can safely target that in the new era. So again, this was the original paper that we published back in 2014, now is almost 8 years ago, where we looked at the role of BCO2 inhibitor venetoclax in acute myeloid leukemia. First of all, we published that BCO2 is highly expressed on both RNA and protein level in primary AML and AML cell lines. So this is the series of large series of primary AML patients from the MLL collection from Thorsten Haferlach. And the beauty is that they had a large number of what they call normal, non-malignant bone marrow. So we could compare the expression to the normal bone marrow. So you can see that most of the AML, regardless of the genetic subsets, they have at least one log higher expression of BCO2 mRNA. So this is log 2 scale, with a few exceptions, perhaps. For example, in version 3 and 821 had low expression of BCO2. So this is a cell line expression of BCO2. They also express other family members, which of course creates mechanisms for potential resistance. And I'll get back to you on that in detail. So we additionally showed that venetoclax effectively kills AML cells, both cell lines and primary cells in vitro. And we showed efficacy in vivo in both cell lines and AML PDX model. Although I have to say we could never really cure mouse containing AML with venetoclax as a single agent, which turned out to be true in clinical trials. So this data provided the rationale for the company to take forward this inhibitor that was already working very well in CLL and was approaching approval in CLL, where it's highly upregulated and it's really critical for the survival of CLL cells, into AML clinical trial. So based on this work, we predicted that we have at least 50% response rate in the relapse refractory AML based on our in vitro killing assays, which turn out to be not quite the same. So this was the first clinical trial of venetoclax single agent. This was the only single agent trial in acute myeloid leukemia relapse refractory setting. We only accrued 32 patients. So the trial was stopped early because of the efficacy of the drug was lower than what we predicted. So the response rate was only about 19%, while it was higher in patients who had IDH1, IDH2 mutations. And I'll discuss that with you later. But still, it was quite a signal because a lot of patients had reduction in the blast count, and the responses happened very quickly within the first 4 weeks. But they didn't last long. So the median time on study was only 2 months, while some patients actually stayed on the study for quite a long time. Again, the responses were enriched in patients with IDH1, 2 mutations, and there was a limited biomarkers done in the study. Tony's lab has performed BHC profiling, which he showed correlated with largely resistance to the drug, where the cells were primed towards BCL-XL and MCL1. And very similar, the flow cytometry, which was done by the company, showed that high BCL2 and low XL MCL1, what they call BCL2 sensitivity index, correlated with the time on study. So this was the first trial. Again, it was stopped because we figured out that the single agent probably cannot get approved as is, with a response rate of only about 19%. Fortunately, this did not stop the further development of this drug, and largely because of the arguments provided by our team, leukemia team, Dr. Contagion, Dr. DiNardo, and others, the company took this trial into the frontline elderly AML patients who were ineligible for intensive chemotherapy. In the past, the outcome of these patients was extremely poor. The only thing available for them was low-intensity chemotherapy, such as hypomethylating agents or low-dose cytarabine. So this trial was aimed to look whether adding venetoclax to this low-intensity chemotherapy will be beneficial. And again, I will spare you all the details of the clinical trial, which went all the way from phase 1 to the phase 3 randomized placebo-controlled trial of AZA, which is this green line, or AZA combined with venetoclax. So what we find out in the phase 1, also reproduced in phase 3, which was large multi-center, nationally, actually worldwide study, which was published by Dr. DiNardo and others in New England Journal of Medicine back in 2020, is that the combination induced a fairly high response rate of 67%. So with the AZA alone, we get only about 20% response rate. But it was not as effective in other studies in relapse refractory AML. But most importantly, there was a survival advantage. So the median overall survival was about 15 months compared to about 9 months with azacitidine. And even though this did not look to be such a win-win, but still these patients were able to tolerate drug very well with very limited toxicities. And it seemed to be a plateau on the curve, although it's only about 30% of the patients that can enjoy long-term perhaps survival and maybe cures. We subsequently took this into the frontline trials with the younger AML, and this was published in JCO last year. And when we combine the venetoclax with chemotherapy, the response rate go up to 90% or even higher. While this is not yet approved, there's clearly improvement in the depth of remissions, and these patients, if they undergo stem cell transplantation, they have a durable survival advantage. But we also figured out that patients are still relapsing and dying. Therefore, they develop acquired resistance, and we focus our efforts in understanding this resistance mechanisms. So again, we went back to lab and did a lot of studies trying to figure out the resistance, which of course is now being addressed by many, many teams across the country and worldwide. So first of all, the question is, who are the patients who benefit most from this particular treatment? So this was a study that we published together with Andrew Way from Melbourne, Australia. So we essentially combined patients treated at our 2 institutions which were the primary 2 large centers accruing to the study. So we combined patients treated with both hypomethylating agents or low-dose cytarabine and venetoclax. And we divided them in those who enjoy long-term durable remission more than a year, or those who achieved remission and then relapse, or those who were primary refractory. So looking at this first column, durable remissions, this was mainly looking at the mutation analysis. You can appreciate that patients who had NPM1 mutations or IDH1, IDH2 mutations were enriched in this category. And perhaps you remember that IDH1, IDH2 were also enriched in the single agent venetoclax trial. So why that is the case, I have to say that when the original trial was running with a single agent venetoclax, this paper came out from Ravi Majeta's lab, where they showed that the oncometabolite called 2-hydroxyglutarate, which is produced in IDH1-2 mutated patients, it inhibited the enzyme called COX, cytochrome C oxidase, that is located at the outside mitochondrial membrane. And therefore, cells to survive, they have to depend on BCL2, and they become synthetically dependent on BCL2 for survival. Therefore, if you use BCL2 inhibitor, the cells are essentially dying very quickly at low IC50 compared with IDH wild type cells. They also show this not just with the drug, but by the RNA screen, where the BCL2 sensitivity came up as a top target. So this explains in part why IDH1-2 mutated AML are as sensitive. And this actually borne out in this randomized phase 3 study. It's called VIALE-A study. So this shows for you the response rate in IDH1 and IDH2 mutated AML comparing aza-ven versus aza placebo. So you can see that the response rates are way higher, especially for IDH2 mutated patients, and we don't quite understand that, except of course IDH2 is located in mitochondria, IDH1 is cytosolic enzyme. But looking at the survival, this is particularly impressive. So you can see that these patients are having truly a long-term survival, and perhaps again, some of them may be cured. In fact, more than 50% perhaps enjoy a long-term survival benefit. As a follow-up to that study, we collaborated with Dr. Seri in France, and he published this very extensive paper looking at the OX40 dependency of IDH1 mutated AML. So he discovered, similar to some of the prior work, that IDH mutation causes upregulation of the oxalophosphorylation and ETC1 electron transport chain 1 complexes. So the mechanism that they identified was again through this oncometabolite 2-HG that affects methylation, which was known before, but they identify specific targets, CPT1 and other fatty acid oxidation genes, as being upregulated. So they showed that the IDH1 mutated AML, they have a high activity of the OX40 and the electron transport chain. And it was already known that actually BCL2 inhibitors can inhibit the OX40 through mechanism which is not quite fully understood. What happens if you treat cells with IDH1 or IDH2 inhibitors? Of course, we have this drug now approved for AML therapy. So you have a reduction of 2-HG, but cells very quickly adopt and they upregulate the same pathway, fatty acid oxidation, through different way. This is molecular mechanisms that they identified. And they again have high OX40, even though they benefit from IDH inhibition. So basically what they show, the combination of IDH inhibitors and BCL2 inhibitors or other OX40 inhibitors was highly beneficial and resulted in extended survival in the mouse studies. So based on this and in some other data, Dr. DiNardo, myself, and others have designed this clinical trial where we combined the IDH inhibitor ivosidenib with venetoclax, and we later added azacitidine because we know that there was a synergy with venetoclax in this trial. So it's a complicated scheme. I'm not going to go through that, and there's several dose levels that we exploited. These patients are fairly rare, so the study is still ongoing. But looking at the response rate, again, these are two different dose levels. This is combination with AZA. You get 67 at the low dose level, but 100% response rate in the newly diagnosed patients, clearly higher than what you get with aza-ven or with AZA-IDH1 inhibitor. So this trial is ongoing. The responses seem to be durable. And we'll collaborate with Paresh Vyas from Oxford. He presented his data yesterday at the Grand Round. Some of them may have listened to his talk, where he does very extensive genomic analysis. And my lab with April Zhang is doing the site of analysis on this trial, trying to identify what happens to the clone with respect to the leukemia stem cells and differentiated cells. So this is an example of one patient site of analysis who had large fraction of CD34 positive cells prior to the treatment. It actually clustered in 4 different clusters based on some of the other markers included in the panel. And after, I think this was 3 months of treatment, there was very small population that was remaining. You can see here 0.4, 0.3% population 0 and 1 compared to this, and the non-existing the other 2 population. So in the panel that we have includes large BCL2 profiling and some other signaling pathways. So looking at this residual population, even though it's very small, it actually showed upregulation of BCL-XL and MCL1. So we'll have to see whether it will be eliminated with long-term treatment or whether it actually will grow out and result in the relapse. But generally speaking, you can appreciate a large reduction of the leukemia stem cell clone with this doubles therapy that we utilized. Now, going back to the resistance, so that was a sensitive subset, IDH mutated AML. What about the resistant subset? So these are the data from an original single agent trial of venetoclax, where we did genomic analysis of patients. And we divided them as those who had no activity, no blast reduction, or those who had blast reduction, and look at the mutations. It was actually whole exome sequencing, but we didn't have normal control, so we did not identify new mutations, but we could identify known mutations. So again, you can see that IDH1/2 was highly enriched in those who had response. But then there was these 3 patients who still had IDH1/2 mutation, but did not respond. And looking at the concomitant mutations, we identify FLT3-ITD and PTPN11 as being co-expressed in the same clone that had IDH1/2 mutation. But that was most interesting is that patients who initially had response and then they relapsed, we found the same 2 types of mutations, what we call them signaling mutations in FLT3-ITD and PTPN11. So based on this very small study, we concluded that this mutation will correlate with the primary refractoriness, but also cause secondary progression to venetoclax-based therapy. And this correlated with the time on study. Of course, our attention immediately was drawn to the FLT3 mutation because it's very well mutation in AML, and about 30% of AML patients have this mutation. And we do have specific inhibitors that target this mutation. We subsequently looked at the combination trial. This is, again, the same study that I mentioned before, ven-HMA or ven-low-dose cytarabine. And again, we found that this FLT3 mutations or mutation in the RAS pathway that included the PTPN11 in some cases were enriched in patients who either relapsed or those who were primary refractory. And actually in that study, my lab has done a single cell DNA sequencing, and we showed that this mutant clone with FLT3-ITD was not eliminated fully and then coming back at the time of relapse. And the same here for another 2 patients. So again, going back to the biology of the FLT3 mutation, so they are sitting on their cellular membrane, and what is known about them that they are signaling through a number of the downstream signaling pathways such as RAS, PS3 kinase, PIM, NF-κB stats. And what happens downstream is that one of the key family member that's being upregulated is MCL1. So MCL1 is a known resistance factor to BCL2 inhibition that we identified way back. So we suspected that this was a reason why these patients actually progressed. We also hypothesized that if we block this upstream using some of these inhibitors, that some of them are already approved, such as gilteritinib, midostaurin, sorafenib is used off-label, and quizartinib we think is about to be approved, then we can restore the sensitivity to venetoclax. So we also developed the cell line that was resistant to venetoclax. This is ITD carrying cell line. And what we found that it has a large upregulation of MCL1 protein at the time of resistance. And if you use either MCL1 inhibitor or siRNA, you can largely sensitize to the killing by these agents. But again, MCL1 inhibitors are still early in their clinical development. So we used instead the FLT3 inhibitor as upstream regulator of MCL1. And we do see upregulation of MCL1 also in primary samples. So this is again site of analysis from the HMA Venn trial. This is a complicated heat map of the baseline AML sample with a different subset of cells, and this large cluster is defined as leukemia stem progenitor cells. So it has some response down to 4%, but there is clearly residual population remaining, and then there was a relapse. And by our site of analysis, we identified actually upregulation of both BCL2 and MCL1 at the time of relapse. And we found some signaling pathways being upregulated, perhaps driving this MCL1 increase. So again, we combined, in this case, quizartinib, one of the FLT3 inhibitors, with venetoclax, and just looking at the cell line, we showed strong synergy in vitro killing. We also showed, as expected, that quizartinib would inhibit FLT3 signaling. You see here downregulation of phospho-FLT3, phospho-STAT5, and it partially reduced MCL1. I have to say not completely. So we do have a project looking at the, in fact, combination of MCL1 and FLT3 inhibitors. We also showed this by the BHD profiling that would prime cells towards BCL2 inhibition. And this is one mouse model. Granted, this was subcutaneous mouse model, but we subsequently published systemic mouse model where the mice were quite resistant to the venetoclax alone, similar to our clinical data. They were sensitive to quizartinib, but the combination eliminated the leukemia. So based on this and some other evidence, we developed a program where we combined venetoclax with FLT3 inhibitors, and that also went into clinical trials. So these trials are led by Dr. Davar from our institution and a number of other colleagues across the country. So this is a study that was presented several times. This is relapse refractory AML that have FLT3 mutated disease, and they were treated in combination of venetoclax and the gilteritinib. So again, looking at the response rate, overall response rate is about 75 to 80%. And most importantly, that these patients have already failed prior TKI, tyrosine kinase inhibitors, and still we can regain the response by adding venetoclax. And if these patients are able to go towards stem cell transplantation, they're actually doing very well as far as the overall survival. So this study is about to complete, and the formal phase 2 studies will be starting, hoping that we can get approval of the strategy in AML. We also moved that into the frontline therapy, which appears to be highly effective, but I don't have time to show you that data today. So now moving forward, what about other mechanisms of resistance? Again, this is the same data that I showed you before. And I talked to you about the signaling mutations, which are down here. But there was a much larger cluster of resistance that was identified very early on. And these were patients who had complex karyotype deletion of 17p that has p53 gene and p53 mutations. So we looked at that in detail in this study and subsequent study. And this is our in-house study where we actually intensified hypomethylating agent. And this looking at the survival of patients who did not have or who had p53 mutation. So patients who had p53 mutation, they only lived about 5 months. So this was really disappointing, knowing that BCL2 inhibition is downstream of p53. So this was quite unexpected that we see such a resistance. So essentially, this is the same that we get with hypomethylating agents. And I would say that this is the biggest unmet need right now in AML therapy. Looking at the genomics, different mutation clearance, so you can see that patients who achieve CR, there is a clearance of the majority of the mutations. This work was done by John Welch under the NIH funding that we have together on the clinical trial. But this patient had p53 mutation, which absolutely did not change here, the blue curve, after the first cycle of therapy, even though the blast count goes down, but the clone survives and leads to very rapid progression of the disease. So we decided to go after that with looking at the immune therapy, because we knew that immune therapy may work regardless of p53 mutation. And at the same time, there were emerging data that targeting CD47 don't eat me signal is highly effective in patients with P50 mutant AML. So there's antibody that was developed by Weissman, Ravi Majedi, and others, and the company was 47. It's called now magrolimab, anti-CD47 monoclonal antibody that blocks this don't eat me signal. So very briefly going through biology. So again, this is a macrophage and this is a cancer cell. So macrophage is expected to eat phagocytose cancer cells if there is any chemotherapy, let's say, that damages the cancer cell. But then if the cells express this CD47, there's a block of this process, and therefore leukemia cells in this case will survive without phagocytosis. Now, if you have this magrolimab, which is antibody shown here, it would coat the cell surface of leukemia cells, and now macrophages are actually able to engulf and phagocytose as shown here. These are the red macrophages. These are the data from company, and you can see that after blocking 47, there's a lot of phagocytosis going on. So it was already shown that in MDS and also in AML, combination of azacitidine and this antibody is highly effective in P50 mutated AML. So 6-month survival was 90%. If you remember in our study, most patients died before the 6 months mark, unfortunately. So we asked the question, what if we add venetoclax? We know that venetoclax by itself is not effective against P50 mutated AML, but what if we combine with the magrolimab? So what was the rationale for that? We know that venetoclax, in addition to inducing cell death, it also induces, typical PS and Nexin exposure, which is a classical signal for the phagocytosis. But perhaps there are other signals that we are still investigating currently. So we have looked at the combination of 47 and ven-aza in the in vitro culture system, of which we established looking at the macrophages and target cells labeled with the calcine and identifying macrophages by flow cytometry. The ones that would phagocytose, they have a double positivity. So we compared the leukemia cell line, and which was parental resistant to venetoclax or P50 mutated. So the labels here is in blue is DMSO, and in red is cells pre-exposed to ven-aza, venetoclax and azacitidine. And here the second column is with the anti-CD47 targeting. So you can see that essentially in all conditions, aza-ven by itself is actually quite potent inducer of phagocytosis. This was something that we did not even anticipate. And perhaps there was some increase with the anti-CD47 targeting. But again, these are in vitro systems, which may or may not be true. So we took this forward into the in vivo studies. So we designed this clinical study where we injected the AML PDX sample into NSG mice. And these mice will receive treatment with azacitidine, venetoclax, or 5F9, which was delivered at the schedule IP for 3 weeks. So this is looking at the tumor burden, and this is perhaps survival, which is most apparent. The treatment was stopped after 50 days. But you can see that both 5F9 and ven-aza can induce extension of survival, but the combination is essentially doubling that. And again, we stopped treatment, which we wouldn't do in clinic. We did another PDX sample, which was totally refractory to ven-aza, but it was sensitive to 5F9. But still, the combination was giving us the median overall survival of 385 days, so way longer than the magrolimab alone. So we continue this study. We are looking at specific pro-phagocytic signals that are induced by venetoclax. We are looking at the macrophages, and of course, this concept was taken into clinical trial, perhaps even before this data were generated or in parallel with that. And this was again led by Dr. Naval Davar from our institution and presented at ASH this year. So at what we had at ASH, now we have much more patients enrolled in this study. In the frontline cohort, 14 were P50 mutated and 11 had wild type P53. So first of all, the response rate in the wild type P50 is 100%. So it's it can go higher than that. So it's very active. If you remember with aza, it was about 70%, and with the aza, magrolimab was also about 65%, a little bit lower. Now, in the P50 mutated, there is still very significant response rate, and we're trying to get all these patients towards stem cell transplantation. We do not have yet long-term outcomes, so we really need to look at the survival. In the relapse refractory patients, those who did not have prior venetoclax, they also did very well. But those who had a prior ven, they didn't do very well. So we were unable to conquer the resistance to ven with this approach. But still, this is a very exciting study that is continuing, and myself and Dr. Hussein Abbas and the others will be doing in-depth single-cell genomics and proteomic studies trying to identify signals in both leukemic cells and macrophages. So the last part of my talk, and then we'll get to questions, is about the role of BCL-XL. So I mentioned to you before that the targeting of BCL-XL stumbled previously because of the inhibitor having the toxicity against platelets. So essentially, patients developed, it was taken into clinical trials with navitoclax as a drug, and patients were developing thrombocytopenia, which required dose reductions, and therefore, perhaps the therapeutic window was limited. But we try to understand first, does BCL-XL play a role in lymphoblastic leukemia or myeloid leukemia? So starting with ALL, so this was a paper that published again from Tony Letai's group again back in 2014, where they looked at the T-ALL subset. So I don't have time to show you data on the BLL that are highly BCL2 dependent, and we are doing the clinical trial with venetoclax in that subset that appears to be preliminary highly positive. But the TLL, there are again two different subsets within that TLL, which originates from early T cell. One is called so-called ETP, early T cell precursor AML, and then there's a typical more mature ALL. So these patients have somewhat stem cell biology. They have co-expression of the myeloid markers, and the origin of leukemia is known to be early. So if you look at the expression of BCL2 and BCL-XL, there's a very interesting switch here. So the ETP ALL have BCL2, and the little BCL-XL, while TLL others, they have actually high expression of BCL-XL, and some of them also will have BCL2. So Tony's lab has shown this both by BHC profiling, where ETP-ALL are BCL2 primed and the TLL are BCL-XL primed. And as a consequence, so you can see that if mice are treated with the venetoclax BCL2 inhibitor, in ETP, there's very nice response, but there's no response in the TLL other, while dual BCL2-XL inhibitor has actually very nice reduction of the tumor burden. So we looked at this a little bit more in detail, and this is work which was done by students, Nas and Julia in my lab. So we first look at the expression of BCL2-XL, just looking at the cell line profile. So these are BLL, pH-like ALL, BLL, and TLL cell line. So if you look at BCL2 expression, the BLL cells have high expression of BCL2, but TLL have actually low expression. And again, the opposite is true, which fits very well into the first paper. I showed that TLL others have high expression of BCL-XL, and perhaps also this pH-like ALL. MCL1 is sort of variably expressed in the subset. We also have done the BHC profiling, and so BHC profiling is based on the exposure of the cells to the specific peptide that have specificity against the different BCL2 family members. And therefore, you can dissect if the cell actually functionally depends on BCL2-XL, MCL1, or others by measuring cytochrome C release by flow cytometry. So this is a bit complicated slide, but there are multiple different peptides that determine just regular priming. So generally speaking, these are BLL and TLL. The ALL cells are primed towards these peptides. But then looking specifically at the BCL2, by using bad peptide or BCL2 inhibitor, you can see that BLL have perhaps higher priming against for this NALM6 cells that don't have bags. But TLL have low priming. But also looking at BCL-XL by specific peptide HRK, only TLL are primed towards BCL-XL. So again, suggesting that in TLL, BCL-XL actually plays a big role. So then we compared the responses to the BCL2 inhibitor or to the dual BCL2 XL inhibitor. This was a compound that actually went into clinical trials, so-called navitoclax. So just looking roughly at these two graphs, you can see that only few cell lines, in fact, respond to BCL2 inhibition. But almost all cell lines, again, except for this one, would respond to the dual BCL2 XL inhibition. So again, pointing out to the fact that the BCL-XL is important in TLL. So how can we target BCL-XL safely? So there are two approaches that right now are both made to clinical trial. So first is AstraZeneca compound called AZD0466 is a dual inhibitor of BCL2 BCL-XL. It was published last year in Communication Biology by the company. And it's packaged into the nanomedicine liposomal structure, which was shown to prevent the bone marrow toxicity in their preclinical studies. You can refer to this paper. So we looked at the activity of this compound in the TLL and BLL cell line. And this is, again, comparing venetoclax or this AZD in the solid lines. So you can see in BLL, 3 cell lines, they have roughly the same response to both of them. But in the TLL, PH-like ALL, the AZD works much better with the shift of the curve to the left. We took this forward into the in vivo studies, and while ABT9 had no activity, this compound actually extended survival of the mice, and there was no decrease in the platelet count. So this drug right now is in the clinical trial that we're also leading in our institution is multi-center trial, both in AML and ALL. Right now is a phase 1 dose escalation trial, so we have to see how that plays out. But so far it appears to be safe and does not drop platelets. But we're also very excited about the other approach that I will describe to you. So using the PROTAC. We collaborate with Dr. Da-Hong Zhou from University of Florida, who designed the first BCL-XL PROTAC, which was linked to the VHL ubiquitin ligase. So this is the schematics of how it works. So it has the ligand, and the ligand in this case is actually the same drug, navitoclax. So I've optimized that drug. Then there's a linker that links it to the VHL complex and then brings in this ubiquitin ligase, a large complex that degrades BCL-XL. So what is the rationale using the PROTAC rather than small molecule inhibitor that seem to work very well? So again, the advantage is that this particular ubiquitin ligase, it's not expressed in platelets, but it is expressed in tumor cells, including leukemia. So essentially it would degrade BCL-XL in tumors, but you will spare some of the normal tissues. And this is exactly what they showed. This is a joint paper that was published in Nature Medicine 2019. So again, this is the structure of this large compound. And if you look at the platelet, there's no change in BCL-XL level. But if you look at the TLL cell line, this is a drug called DT2216. There's very nice degradation of BCL-XL. This is the dual BCL2 XL inhibitor, which serves as a ligand here. You can see, of course, that it does not drop the level because it just inhibits the function. And that translates into very nice killing, actually more active killing of this TLL cell line, but essentially no toxicity against platelets. So our lab has done the studies in the TLL PDX samples injected in the mice. And again, here, this DT alone, it has activity on its own, and it works better when combined with the chemotherapy. And most importantly, we show that indeed BCL-XL was nicely degraded, and also there was no change in the platelet count or body weight. So this therapy is very safe and is actually now transitioned to the clinical trials. So what are other subsets? So of course, my lab is most interested in AML. So we were wondering if there's any AML subset that also might depend on BCL-XL, not just T-ALL. So these are some data that we generated in collaboration with Dr. Yu from Jiang Yu from St. Jude. So he's bioinformatician by training, and he designed this program he calls it NetBit, which is essentially a bioinformatic way to analyze transcriptome data. So the nice thing is that rather than looking at the expression of the gene or transcript, this program actually infers activity of this particular protein and can identify what he calls as a hidden driver. So these are some of the facts that he has published previously, and this was previously reported in several papers. And this last paper that again was collaboration with our team identified, for example, LCK and BSO2 dependency in TELL. So we asked Dr. Yu to look at some of the available gene expression data sets and specifically at the AML and to identify which subsets might have higher BCL-XL activity. So this is what he came up with. So this was a pediatric data set that also included some adult ALL, included AML, mixed phenotype ALL. This is a new subset I'm not going to go into, and TLL. So I would like to point your attention to these two boxes. So this is a TLL, this BCL-XL, which the gene name is BCL2L1. So you can see that TLL has a higher activity. So it's not expression. It correlates with expression, but this is really like functional readout based on a lot of database searching. But the highest was actually in the subset of acute myeloid leukemia, which is called acute erythroblastic leukemia or AEL. And the former classification is AML-M6. So these are the tumors that arise from the erythroid progenitors. Now, it was also interesting that BCL2 had exactly the opposite pattern. So these are your typical AML, which is highly BCL2 dependent and has high BCL2 activity. This is a TLL, which can be codependent on BCL2, as I showed you previously, but the AEL was essentially BCL2 not dependent. And there was very strong inverse correlation between BCL2 and BCL-XL looking at this gene expression set. So we were interested whether really this AEL, which has extremely poor prognosis, has a P50 mutation, would be sensitive to the BCL-XL inhibition. So first we looked at some other data sets with the help of Xiaoping Su here at Bioinformatics. So these are TCG and BEAT AML data sets. And again, the same was true that AML-M6 have a sky-high BCL-XL activity, but essentially no BCL2. And also the M7 was the same pattern. The same was seen in the BEAT AML. This is for the M7 megakaryocytic leukemia. So we then reached out to Charles Mulligan and Ilaria at St. Jude, who have compiled a large series of AL cases and published this in several reports. And they mined their data looking at BCL2L1. Again, it was sky-high BCL2L1 expression in this subset. And these are the data which are not yet published. So again, this was from the single-cell RNA sequencing of one AL case. And this again was looking at the activity of BCL-XL or BCL2. So the erythroid, there is heterogeneity as you could expect within the AML sample, and the erythroid progenitors have very high BCL-XL activity, but very low BCL2. But then some of the stem cells, and we don't really know yet if these are leukemic or normal cells, they have opposite, they have high BCL2 activity. There was correlation between BCL-XL and GATA1, which is a known transcription factor for the erythroid lineage. We then look back at some of the reported binding data by the cut and run using GATA1. And we identified that GATA1, in fact, can directly bind to the BCL-XL promoter. So that suggests that in erythroid lineages, the BCL-XL is the direct target of the GATA1. So what about activity of the compound? So this, we looked here at this DT, which is BCL-XL inhibitor, and this is a novel inhibitor. I'll tell you in a second. And we identified that the cells that were resistant to venetoclax, they were highly sensitive to the BCL-XL inhibition. This red line is actually dual BCL2 BCL-XL product. So we're taking this forward into more studies and doing in vivo experiments with these agents. Since then, they have also came up with a dual BCL2 BCL-XL product, which they named 753B, by modifying the binding to this VHL and BCL2 pocket, looking at specific ligands. This work was recently published. So now we have a drug that can in fact degrade both BCL-XL and BCL2, and we expect that it will have pretty broad activity in AML. In fact, that's what we showed. It has very nice activity here in red and different cell lines. And in patient samples, it's more sensitive looking at the IC50 compared to the venetoclax, and that has a similar activity to the BCL2 BCL-XL inhibitor. So I'm running a little bit out of time, but we also did some in vivo studies and showed some extension of survival. So we're working now very diligently on this dual degrader. And the last kind of different aspect of the BCL-XL inhibition is that it controls the survival of the senescent cells. So again, it was shown that chemotherapy can induce senescence, and the senescent cells can escape and cause relapse later on. So in fact, we tried to identify this, if this is a fact in AML, and we showed that cells that have senescence by this marker C12FDG, they have high levels of BCL-XL, and they're very sensitive to the inhibitor of BCL-XL 753B here. They reduce the FDG positivity, and they reduce the cytokine production. So again, this is very early on, but this is something we're interested to explore it further. So it's my last slide. So what I showed to you is that AML cell survival is BCL2 dependent, but I didn't talk about MCL1, but it's certainly codependent on other family members in specific subsets or after the treatment with venetoclax. So we're doing multiple combinations of venetoclax, of course, with chemotherapy as a backbone, but also with targeted agents and immune therapies. I showed you only one example out of several ones. We found that subsets of TLL and also AML are BCL-XL dependent, and we're looking at the BCL2 BCL-XL targeting using PROTACs, which actually spare platelet toxicity. We're also interested to learn more about the senescence and how that plays out in the resistance to the chemotherapy. So this is my very last slide. Of course, this work would not be possible without many people who contributed to this from my lab and Michael Andreev's lab. A lot of clinical investigators who are listed here in the Department of Leukemia, research nurses, biostatisticians, and a lot of collaborators with the academic institutions. I mentioned all of them, and the companies and the funding. So I'll stop here, and I'll be happy to take questions in the last 7 minutes.
Thank you very much, Marina. Wonderful talk, a lot of clinical applications. So let's start with the questions. So which analyses help to identify the clearing of mutation, P53 AML treated?
So we used both analysis that was done by the clinical laboratory. We have the 81 gene panel that is done in molecular lab at MD Anderson. So we looked at that, but also we have done some of the single cell DNA sequencing for some specific mutations that we are looking at. But as far as P53, that was mainly the clinical lab that was performing this.
Thank you. Another question regarding the anti-CD47 trial. Can you comment on the mechanism of synergies? Could it be that the effect of HMA is to increase the number of functional macrophages in differentiating leukemia blast to myeloid lineage?
Yeah, I think that's a great question. And it's difficult to dissect in the lab. Which is why we are really looking forward for analysis of the clinical trial. So in the clinical trial, we're going to be doing both single cell RNA sequencing and also the cytoph analysis, looking at the macrophages and leukemia cells before and after therapy. We're also thinking of isolating, FACS sorting macrophages with the help of Sabrina, and looking at some of the signals that may be altered by this therapy in the macrophages. So the problem, of course, that this is already kind of combination trial, right? So we may need to find some historical controls to really figure out what each of the drugs are doing. We don't really have AZA treated patients anymore, but in any case, within that clinical trial, we're going to identify that. But in the lab, we actually focus more on the venetoclax right now. So we're doing experiments looking at the proteomic analysis of cells treated with venetoclax to identify some of these prophagocytic signals. And we will also do the CRISPR screen both on the leukemia cells and macrophages untreated or treated with venetoclax, trying to figure out if the macrophages are in fact having some distinct signals by this therapy that would increase their phagocytic activity. We may look into AZE as well, as was pointed out. So thank you for this question.
Thank you. Another question. Did you see any correlation with SRFS2 expression and sensitivity to BCL2?
So not expression, but SRSF2 is one of the splicing mutation that is common in AML. And I didn't point out on my slide, but in the original trial, we showed that patients who had splicing mutation, they were also more sensitive to the venetoclax single agent therapy. But they commonly had co-mutations in IDH1/2, so it was difficult to figure out what really drives the sensitivity. Dr. DiNardo later with Curtis analyzed the combination trial, and they also showed that these patients with splicing factor mutations, they are more sensitive to combination therapy. So there's a clinical signal. We haven't really dissected this in the lab, what may drive the responses. I think some of you may have heard recent talk by Omar Abdelwahab from Sloan Kettering, and he identified some of the factors that may play a role in that. So I think we will learn from his lab what really drives the sensitivity. But yes, in general, these patients are more sensitive.
Thank you. Next one. What is the role of 753B in BCLX PROTAC treatment?
So I have to apologize, I went too quickly. So there are two PROTACs. So one is the original compound is BCLXL only. It's called GT2216. So even though the binding ligand is the same, so it's based on the navitoclax, so it does bind to BCL2. But what Dr. Zhou's lab has identified that the way it binds, it does not bring ubiquitin ligase to target BCL2. So it only degrades BCLXL. So after that, they did a lot of structural biology, and they identified the license in the BCL2 and BCLXL that are important for bringing in this VHL ubiquitin ligase complex. So by doing so, they were able to come up with the second generation compound that they named 753B that is now more potent BCLXL degrader, but can also degrade BCL2. So this is the new compound. It's being, it's still preclinical compound, but essentially is a product that can degrade both BCL2 and BCLXL. Although I have to say that in our studies, it does degrade BCL2 in majority of the cells, but not in all. So there's something that we don't quite understand. It degrades BCLXL in all cells we looked at, but some cells, they do not degrade BCL2, and we don't quite know why.
Thank you. And now one more question. So BCL2, it's regulated by a lot of factors, translocation, methylation, microRNAs, and others. Did you check on your responders versus non-responders? What's happened with these factors to induce upregulation of BCL2 in order to predict or in order to understand before the start of the therapy if the patient answers or not?
So the short answer is no, we haven't. So we actually do not know why in AML, unlike CLL, BCL2 is highly expressed, and why the AML cells are primed towards BCL2, whether it's microRNA or some of these other transcription factors that were mentioned. So no, we haven't really done the studies looking at what is regulating upstream. So we mainly look at the correlation between the expression of the BCL2 itself and response. And then we looked at what happens after treatment. But the one study that we actually have in press is that we try to, again, look at the resistance to BCL2 inhibition. So we established these paired cell lines where they became BCL2 inhibitor resistant. And we did extensive proteomic methylation analysis of the cells. So we identified that there were a major switch in methylation pattern after cells became resistant, for one. And second, we identified upregulation of the MAP kinase pathway on the RNA level. Even though we don't know what drives that upstream, we think it's RAS, but we don't really have data to support this. But MAP kinase was largely upregulated on the gene expression level, on the protein level. And when we did in the lab treatment with MAP kinase inhibitor, they were able to rescue the resistance to venetoclax. So that happens as adaptation to the therapy. My lab has not looked at what sort of drives the original expression of BCL2.
Thank you very much. So Marina, thank you for your wonderful talk. Let's see one more. Okay, thank you for this wonderful talk and for answering the questions. So the people really enjoy a lot of your talk as myself. Thank you very much. Wonderful for all of us, and good luck with your work and good luck with your research, and to all attendees, good luck, enjoy science and enjoy life. Thank you very much.
Thanks everybody who joined.
Thank you again, Marina. Thank you. Thank you again.
Bye.
Thank you, Emily. Thank you.